Welcome to the Deep Dive. We're your shortcut to getting informed on some really complex topics.
And today, we're looking at something quite remarkable in neurodegenerative disease research.
Absolutely. We're talking about amyotrophic lateral sclerosis, ALS, and frontotemporal dementia, or FTD.
Specifically, how a tiny change, a mutation in the velocin-containing protein gene, VCP, has these huge downstream effects.
It messes with our cells' powerhouses, the mitochondria, in a really unexpected way.
Almost forces them into this kind of self-destructive overdrive.
So our mission for you today is to unpack this. We want to show you how this research reveals potential new ways to maybe treat these conditions.
Okay, so let's start with VCP itself. What does it normally do?
Well, think of it like a master mechanic inside every cell. It's involved in so much DNA repair, getting rid of faulty proteins.
It's a really vital protein, makes up, nearly 1% of all the protein in our cells?
Exactly. So it's a big deal. And when you have certain mutations, even subtle ones, the impact is huge.
Right. We know these mutations are linked to multisystem proteinopathy 1, which is this rare protein buildup disorder.
But also, crucially, to both ALS and FTD. And we're increasingly seeing those two as points on a kind of shared disease spectrum.
Okay, so the link is there. Now, what did the researchers actually find when they looked at cells with this mutation? They used neuroblastoma cells, right?
Yeah, human neuroblastoma cells, engineered with a specific VCP mutation found in patients VCPR191Q.
And the first thing they noticed was the mitochondria.
That's right. They were strikingly enlarged. I mean, almost 27% bigger than normal mitochondria in control cells.
Wow, okay. Bigger powerhouses. But does bigger mean better here?
Well, that's the puzzle. Because even though they were bigger, their battery charge, the mitochondrial membrane potential was significantly lower, depolarized.
So they're struggling, losing that crucial energy gradient needed to, make energy effectively.
Exactly. It's like the lights are dimming even though the power station looks bigger.
Which seems counterintuitive. So what were these struggling mitochondria actually doing in terms of activity?
Okay, get this. This is where it gets really interesting and paradoxical.
Despite being depolarized, they found increased oxygen consumption.
Right. And higher activity in the electron transport chain. It's a state they're calling hypermetabolism.
So it's like flooring the gas pedal in a car that's stuck in neutral, burning lots of fuel but not really going anywhere productively.
That's a great analogy. They're revving their engines like crazy, possibly trying to compensate for something.
And this extra activity, this wastefulness, seems tied to a proton leak, right? Across the inner mitochondrial membrane.
Yes, exactly. It's like there's a short circuit, letting the energy dissipate as heat instead of being properly harnessed.
Okay, so we have enlarged, depolarized mitochondria that are paradoxically hypermetabolic because of a proton leak. So the next question is, what's causing the leak?
What's essentially poking holes in the mitochondrial battery?
Well, the research points pretty strongly towards something called the mitochondrial permeability transition pore, or MPTP.
The MPTP, that's like a regulated gate or channel on the mitochondria.
Precisely. And when it opens too much or stays open too long, it lets essential molecules leak out.
Causing the depolarization.
Exactly.
Yeah.
And likely triggering that compensatory hypermetabolism we just talked about.
And did they test this? Could they show the MPTP was involved?
They did. They used inhibitors of the MPTP, and guess what? The mitochondrial membrane potential, that battery charge, was significantly restored.
Wow. Okay, so the MPTP is definitely a key player here. It's the source of the leak. But what's pushing it open in these VCP mutant cells? What's the trigger?
Right. What's the upstream signal?
Yeah.
The evidence is compelling, and it points to calcium, specifically mitochondrial calcium overload.
Calcium overload inside the mitochondria.
Uh-huh. The cells with the VCP mutation had higher baseline levels of calcium inside their mitochondria compared to normal cells.
And they could take up more too.
Yes, they also showed an increased capacity to sequester or take up even more calcium when the cell was stimulated.
So they're primed to accumulate calcium, and they do. And is there evidence linking that calcium overload directly to the MPTP opening?
Yes. They found that if they blocked the main channel that lets calcium into the mitochondrial calcium uniporter, or MCU, it significantly reduced MPTP opening and helped restore that membrane potential.
Okay, so the chain seems to be VCP mutation leads to problems handling calcium,
Which leads to calcium overload inside the mitochondria,
which forces open the MPTP gate.
causing the proton leak, the depolarization, and that frantic hypermetabolic state.
Right. So let's connect all these dots. What does this whole cascade mean for you for someone trying to understand ALS and FTD?
Well, it suggests a really compelling new model for how these diseases might develop, at least in cases linked to VCP mutations.
It's not just about protein clumps, maybe.
It seems this mitochondrial dysfunction, this hypermetabolic stress driven by calcium and the MPTP, could be a really important, perhaps early factor.
So the cell tries to adapt with this hypermetabolism,
Right. It might be adaptive initially, but over the long haul, this constant overdrive, this inefficiency, could just wear out the mitochondria faster.
accelerating mitochondrial aging and dysfunction.
Exactly.
Yeah.
And ultimately contributing to the death of neurons that we see in ALS and FTD.
That's quite a pathway. And the exciting part,
The exciting part is that it shines a spotlight on new potential targets for therapies.
like what?
Well, if calcium dysregulation and MPTP opening are key drivers, then maybe we could develop drugs to modulate mitochondrial calcium levels, perhaps block the MCU channel carefully.
Or find ways to stabilize the MPTP, keep it from opening inappropriately.
Precisely. These are now really interesting avenues to explore for potential treatments for ALS and FTD, stemming directly from understanding this specific mitochondrial problem.
It really is incredible how understanding this one mutation's effect on cellular power plants opens up whole new ways of thinking about tackling these awful diseases.
Absolutely. You can now appreciate just how intricate things are inside our cells and how one problem can cascade.
So a final thought to leave you with.
Yeah, if this hypermetabolic state really is an early, maybe adaptive response, could intervening then, maybe by carefully managing that mitochondrial calcium, be a way to prevent the later stages? Could we stop the neurodegeneration before it truly takes hold?
A really provocative question. Something to think about. Thanks for joining us on the Deep Dive.